I am Payal Predarshini, PGT Biotechnology, Kendriya Vidyalaya number 3, Delhi Cant, and today I am going to make you learn about structure of DNA and the function of DNA. In this session you will learn how the DNA has been synthesized, what are the different components of the DNA, how those components are being bonded with each other, and what is the function of the DNA. So, as we know कि DNA is blueprint of life. What is the meaning of blueprint? Blueprint means whatever the cell has to perform their function, whatever the cell has to perform their activity, that is under the control of the DNA. That means DNA directs the cell when the cell have to divide, when the cells have to undergo reproduction and other activities. That is why DNA is called the blueprint of life. Let us know something about the journey of the DNA. Today we know about the DNA because of the contribution of some of the scientists. Let us know some of them. The first of all, the DNA was observed by a great scientist in 1868, Frederick Visser, in the WBC cells of pus, and he named this compound as nuclein. Later on, Erwin Chargaff in 1950 gave the ratio which is also called Chargaff ratio, which states that in a particular DNA fragment, number of purine will always be equal to number of pyrimidine. If suppose there are thousands adenine nucleotide in a particular DNA fragment, then there will be 1000 thymine in that DNA. Similarly, the concentration of guanine will always be equal to concentration of cytosine in a particular DNA fragment. After this, Rosalind Franklin and Maurice Wilkins gave the first picture of DNA with the help of X-ray crystallography. With the help of that, Watson and Crick determined the structure of DNA. And then finally, Watson and Crick in 1953 discovered the structure of DNA, what we learn today as double helical structure. Now, what are the chemical composition of DNA? What are the different chemicals which form the DNA? We will see in the next coming slide. DNA is considered as biomolecule. Why it is considered as biomolecule? Because biomolecules are present in cells and control the different function of the cells. Like that, DNA is also present in the cells and control various activities of the cells. DNA synthesize a protein and protein function various types of activities. DNA is composed of nucleotides. Various types of nucleotides combines together to form the DNA. That is why nucleotide is the monomeric unit of DNA. Now, what are nucleotides? Nucleotides consist of three major components. One is sugar, that is carbohydrate. Second one is nitrogenous base. Third one is phosphate molecule. These three major components combines together to form one nucleotide, and many of the nucleotides combines to form nucleic acid, that may be DNA or RNA. Sugar and nitrogenous base combines together to form nucleoside, and nucleoside combined with phosphate to form nucleotide. And as told you, many of the nucleotides combine to form nucleic acid, which may be DNA or RNA. Now, let us see which sugar makes the nucleotide, or we can say makes the DNA. There is deoxyribose sugar as well as ribose sugar, which constitutes the nucleic acid. As we know, nucleic acid is of two types, DNA and RNA. DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. DNA is having deoxyribose sugar while RNA is having ribose sugar. What is the major difference between these two sugar, ribose sugar and deoxyribose sugar? If you can see in the picture, ribose sugar is having 5 carbon, that is why ribose sugar is considered as pentose sugar. Here are the carbons, carbon number 1, 2, 3, 4 and 5, and this one is deoxyribose sugar, it is also having 5 carbon, that is it is a pentose sugar. The major difference in between these two sugar, ribose and deoxyribose sugar, is at the second number of carbon in ribose sugar there is hydroxyl group, OH group, while in deoxyribose sugar at second number of carbon there is deficiency of one oxygen molecule, only hydrogen is there. That is why this is known as deoxy, it means deficiency of oxygen at the second number of carbon, and this is the major difference between ribose sugar and deoxyribose sugar, and deoxyribose sugar is present in DNA. This is one of the major difference between DNA and RNA. If you can see, these sugars are found in cyclic structure. What is the reason behind this? Why the carbohydrate molecule found in cyclic form? And the answer is formation of hemiacetal and hemiketal. Whenever the alcohol and aldehyde or alcohol and ketone combines together, they form their respective compound which is called hemiacetal or hemiketal. This reaction is very much simple, you can understand over here. The hydrogen of alcohol, this hydrogen will transfer at the oxygen of carbonyl carbon and it will form OH. Similarly, the fourth valency of this carbon will be satisfied by the remaining part of the alcohol, that is R-O, and this form the hemiacetal. The same reaction will be here with the alcohol and ketone. The hydrogen of alcohol will transfer over the oxygen, it will form OH, and the remaining part of the alcohol will combine with the carbon and complete its fourth valency and make the hemiketal. The same reaction is occurring in the carbohydrate also. This OH group will participate in the reaction, the hydrogen will transfer over the oxygen of the carbon and it will make OH group, and remaining part of this carbon will form a ring with this carbon number 1, and because of this hemiacetal formation the carbohydrates are found in cyclic structure. This again you will learn in the biomolecule chapter of class 11th. Next major component of DNA is nitrogenous bases. There are 5 types of nitrogenous bases: cytosine, thymine, uracil, guanine and adenine. Commonly they have been classified into 2 categories, pyrimidine and purine. Pyrimidine includes cytosine, thymine, uracil, and purine includes guanine and adenine. Out of these 5 nitrogenous bases, only 4 are present in the DNA and 4 are present in the RNA. Cytosine, thymine, guanine and adenine are present in DNA. Cytosine, uracil, guanine and adenine are present in RNA. The third major component of the DNA is phosphate molecule. When we talk about DNA, there is just one phosphate in the DNA, this phosphate, but when the nucleotides combines together, when the nucleotides combines to form the DNA, while forming the DNA, while synthesizing the DNA, the 3 phosphate molecules are there in the nucleotides, but while making the bond, 2 of this phosphate will remove from this triphosphate, and because of the breaking of the bond, whatever energies are being released out, that energy is utilized to make phosphodiester bond. With the help of that energy, the one phosphate molecule will combine with the sugar molecule of the nucleic acid. Now let us see how these major components of the DNA or RNA combines together and what are the different bonds. First we will see how the sugar molecule combine with the nitrogenous bases. This is the deoxyribose sugar which is present in the DNA. At the first number of carbon, this nitrogenous base, it may be adenine, cytosine, thymine or guanine. Uracil is not there in the DNA. So, this nitrogenous base having NH group over here, the hydrogen of this NH group will bond with the OH of the first number of carbon of the sugar. They will form the water molecule and the water molecule will release out from there, and the remaining part of the nitrogenous base will attach over the first number of carbon to form the nucleosides. So, combination of sugar molecule and nitrogenous base is called nucleoside, and the bond which combine this together is N-glycosidic linkage. There are 4 types of nucleosides respectively in DNA as well as in RNA. In RNA we have adenosine, we have guanosine, we have cytosine and uridine. Similarly, in DNA we have 2-deoxyadenosine, we have 2-deoxyguanidine, we have 2-deoxycytidine and thymidine. So, these are the 4 types of nucleosides which further combine with the phosphate molecule to form the nucleotide. Here you can see how the nucleosides combine with the phosphate molecule to form nucleotide. This you already know, okay, this sugar plus with the nitrogenous base, this is the nucleoside, and this nucleoside will combine with the phosphoric acid. Here you can see the hydrogen of the phosphoric acid and the alcohol of the 5th number of carbon, they will interact with each other and they will form the water molecule. The water molecule will release out and the remaining phosphoric part will combine at the 5th number of carbon to form one nucleotide, and this bond is called phosphoester bond. It is called phosphoester bond because whenever acid and alcohol reacts with each other, they form ester, and here the acid is phosphoric acid which is reacting with the alcohol. That is why this bond in between the phosphate and the sugar is called phosphodiester bond. Let us see how the nucleoside combine with the phosphate molecule. Here you can see this is the sugar molecule combined with the nitrogenous base to form one nucleoside, and this phosphate molecule will interact with the alcohol of the 5th number of carbon, the hydrogen of phosphate molecule and the alcohol part of the sugar molecule. They interact with each other, they will form water molecule and the water will release out, and the remaining part of the phosphate will combine over the 5th number of carbon and they will form one nucleotide. So, there are 4 types of nucleotides respectively in DNA as well as in RNA. In DNA there are DATPs, DCTPs, DTTPs and DGTPs. Like that in RNA there are ATP, CTP, UTP and GTP. So, these nucleotides combines together to form DNA as well as RNA. These nucleotides combines to form DNA and these nucleotides combines to form RNA. Now, how 2 nucleotides combines together to form the polynucleotide chain? Here you can see this is one of the nucleotide molecule, this is the another DNA nucleotide molecule. The phosphate molecule of the second number of nucleotides will participate in the bond with the 3rd number of carbon of the first nucleotide. Both this hydroxyl molecule will interact with each other, they will form water and the remaining part will combine together, and this bond is called phosphodiester bond because 2 alcohol is participating with 1 acid and they are forming phosphodiester bond. Like that, this third number of OH is free for the bond formation, and this dinucleotide is having two terminals, one is 5 terminal and another is 3 terminal. That is why either it is DNA or RNA, they have 5 prime end and 3 prime end because of the sugar molecule. This is how the nitrogenous bases combines together. Adenine always combine with the thymine and vice versa, thymine will combine always with adenine. Like that, guanine always combine with cytosine and cytosine always combine with the guanine. If we talk about DNA, because DNA is double helical structure, they have 2 strands. So, these 2 strands combines together with the help of these bonds, and this bond is called hydrogen bond. In case of adenine and thymine, there are 2 hydrogen bond, and in case of guanine and cytosine, there are 3 hydrogen bond, and these hydrogen bonds help the 2 strand of the DNA to combine together to form 2 strands of the DNA. This is how the 2 strands of the DNA are being combined with the help of hydrogen bond. So, there is hydrogen bond in between the 2 strands of the DNA, or we can say in between the nitrogenous bases of the DNA. Now, let us see some of the features of the model given by Watson and Crick. The first point related to this model is DNA is double stranded helix. DNA has 2 chains twisted around each other on a common axis, which is right handed helix. You can see in the picture the DNA is double stranded, one this strand and other is this strand, which are twisted with each other. Second point of this model is both these 2 strands have polarity. Polarity means they have 5 prime end and 3 prime end. Both these 2 strands have this 5 prime end and 3 prime end. Next feature of this model is both the strands are anti-parallel. Anti-parallel means one strand will be in 5 prime to 3 prime and another strand will be in 3 prime to 5 prime end. Both the strands cannot have the same ends. That means, if one end is having 5 end in one strand, the another strand there will not be 5 strand, there will be 3 prime end. So, both the strands are anti-parallel in nature and both the strands move in 5 to 3 or 3 to 5 direction. The 2 chains are not identical, but complementary to each other. That means, these two strands are not identical, but both the strands are complementary. Complementary means if adenine nucleotide is there in one of the strand, in another strand there will be thymine nucleotide. Adenine will always combine complementarily with thymine and guanidine always combine with cytidine complementarily. The next point of this Watson and Crick model is each turn of helix is 3.4 nanometer. That means, the complete turn, one complete turn is having the length of 3.4 nanometer with 10 base pairs of the nucleotide. In this complete one turn there are 10 base pairs, and the distance between 2 base pairs, here you can see the distance between 2 base pair is 0.34 nanometer and the width of the DNA is 2 nanometer. Next point of this Watson and Crick model is each strand has hydrophilic deoxyribophosphate backbone on periphery and hydrophobic base stacked inside, means the phosphate and the sugar will be in the outer portion and the bases will be in inner side. Next feature of this model is the two strands of helix are held by hydrogen bond. Means, as I told you earlier, these two strands are bonded together with the hydrogen bond which is present in between the nitrogenous bases. One complete turn of the DNA is having 10 base pairs, and the last Chargaff rule, which was given by Erwin Chargaff, in a particular DNA the concentration of adenine will always be equal to the concentration of thymine, and the concentration of guanine will always be equal to cytosine. If suppose in a particular DNA fragment there are 10,000 adenine, it means there will be 10,000 thymine also. That is called Chargaff's rule. Now, there are some different forms of the DNA. The first form of DNA is bDNA, that is balanced DNA, which was given by Watson and Crick, which I told you in the last slide. Another DNA is aDNA, that is alternate DNA. In the aDNA, in one complete turn there are 11 base pairs, while in bDNA there are 10 base pairs. The third form of DNA is cDNA, having 9 base pairs in one complete turn, which is right handed. Fourth form of DNA is ddNA, having 8 base pairs in one complete turn, and the last form of DNA is zDNA, zigzag DNA, having 12 base pair in one complete turn. These are the 5 different types of DNA forms. Now, let us see the different functions performed by the DNA. First, DNA carries hereditary information. What is the meaning of hereditary information? The molecule which get transformed from one generation to another is DNA. But what is there in the DNA which exactly is having the information related to living organism? And that information is nothing, that is the codes which synthesizes the protein, and protein actually perform the various function in the cells. The next function of DNA is it control various types of metabolic activities of the cells. Whenever cells has to undergo catabolic reaction, the DNA will give the direction, and whenever the cell has to go in anabolic reaction, the DNA will give the direction, and accordingly the cells perform the different functions. The next function of DNA, it enables cells to maintain, grow and divide. The growth and the division of the cells are under the control of the DNA. Next is DNA contribute to variation by undergoing mutation. As we can see, no one is similar in the phenotypically. Phenotypically we are not very much similar, and that is because of the variation in the DNA, that is because of the mutation in the DNA. The next function of the DNA, it brings about differentiation of cell. Differentiation of cells is different from division. Division means a cell divide to form similar type of cells, but in differentiation the cells divide to form different type of cells. They are not similar as that of the parental cell. So, these are the few functions of the DNA. Now, here are some differences between DNA and RNA with 4 types of nitrogenous bases: cytosine, guanine, adenine and uracil. Thymine is not there in RNA, and in DNA is double stranded. They have cytosine, guanine, adenine and thymine bases. So, if you can see, RNA is single stranded and DNA is double stranded. RNA is having AUCG as nitrogenous base and DNA is having ATCG as nitrogenous base. Here are some more differences. As I already told you in the last slides, RNA is having ribose sugar, that is why RNA is called ribonucleic acid, and DNA is having deoxyribose sugar, that is why DNA is called deoxyribonucleic acid. Adenine pairs with uracil in case of transfer RNA, and in case of DNA, adenine pairs with thymine. Regions having complementary nucleotides pairs and form hairpin loop like structure and helicals. The whole RNA is not double stranded, but some of the regions have complementary sequences, that is why they form hairpin like structure. RNA is genetic material in some of the viruses, while DNA is genetic material in all the living organisms except some of the viruses. If we compare the length of the DNA and RNA, the length of RNA is very much shorter as that of the DNA. So, these are the some differences between RNA and DNA. Now, let us answer these questions. Group the following as nitrogenous bases and nucleosides: adenine, cytidine, thymine, guanosine, uracil and cytosine. You have to group them. What are the different nitrogenous bases in this listed molecules and what are the different nucleoside in this listed molecule? And answer is adenine, thymine, uracil and cytosine are the nitrogenous bases, while cytidine and guanosine are nucleoside. It means there is sugar and nitrogenous base. Next question: if a double stranded DNA has 20% of cytosine, calculate the percentage of adenine in the DNA. You can use Chargaff rule for this answer. As we know, according to Chargaff, the concentration of adenine is equal to concentration of thymine and concentration of guanine is equal to concentration of cytosine. In the question we have given 20% of cytosine, it means there will be 20% of guanidine, and this makes 40% of G plus C. So, remaining 60% will be A plus T and this will be equally divided. So, the answer will be 30% adenine will be there in this particular DNA fragment. The next question: if the sequences of one strand of DNA is this, then what will be the complementary sequence? You can use the different points given by the Watson and Crick, that adenine always combined with thymine, thymine always combined with adenine, like that guanine always combined with cytosine in the complementarily. So, answer is this one, you can see in the slide. Next question: which bond exists between two nucleotides and which bond exists between two strands of the DNA? When we talk about two nucleotides which form the strands of the DNA, there is phosphodiester bond in between the alcohol and the acid part of the nucleotides, and if we talk about two strands of the DNA, there is hydrogen bond in between the nitrogenous bases of two strands of the DNA. This question you can answer yourself, and this I can discuss in my next session. Thank you very much. We will meet you soon in the next session.